Welcome back. And in this lesson, we are going to implement your first bioinformatics program that parses the GC content from a breast cancer data file. Through DNA sequencing, we can determine the exact order of nucleotides in an organism's genes, and this allows us to map the DNA sequences of all organisms and connect the gene sequence with the gene function. So first we need to procure the data set to work with. And to do that, I'm going to go to Google. And we are going to type in NCBI. Then we'll go down to where it says nucleotide. And in the search box here, we want the breast cancer, any breast cancer data set. So I'm going to type in BCRA1. And here's one with almost 3 million circular DNA. So we'll go with that one. And we're going to select the FASTA file. And you see all of those nucleotides there. So we'll select this one. We'll go up here to send to. We want the complete record. You could put coding sequences or gene features. And we can put it to the clipboard, analysis tool, collections. But this here time we are going to use file. And we'll keep the FASTA format for right now. There's other formats that you could use. So create file, and that downloads it to my downloads folder on my Mac. So I can now come out of here, and I am going to launch Spyder, which is a scientific Python development environment that allows advanced editing, interactive testing, debugging, and introspection features. Now, if you don't want to use Spyder, or if you don't want to use Anaconda, that's fine. For this particular lesson anyway, you could actually just use your text editor and your console. So if you don't want to download Anaconda and use Spyder, just follow along and use your text editor. Not sure what it would be on a Windows PC, but on a Mac, you would use text edit, and you can open that up if you go to your launch pad, click, go up here to search. If it's not there, type text edit, start, and it'll come right up for you. Okay. So we're going to launch Spyder. I'll start a new file here. So the first thing we want to do is locate that file. You'll see the file in your downloads. It's a FASTA or wherever you downloaded it at. Okay, so before you can pull it up in Spyder, you have to make it a TXT file. So locate that file, copy and paste it, then rename it .txt. So you should have two files, the one that you downloaded and the one that you renamed with a TXT extension. So the first thing you want to do is create a variable object to attach to that file so you can use it. So we'll call it gene equals open sequence .txt and we're not going to write to it, we just want to read it, so we'll give it read permissions with an R. Done deal. Okay. Next, we want to set the value of each nucleotide to 0. So let's do that now. So you'll find a header line in that FASTA file as well as the TXT file, and that would draw a concern for an error. So to get by that, we are just going to skip that line, and to do that, we'll go gene .read line, and that skip that line for us. Okay. Now what we need to do is create a for loop to iterate through the rest of that data set. So let's do that now. Next, we'll get and print the GC ratio. And we'll do the same with the AT nucleobase. So let's give it a run. And you see we have 625 guanines, 630,000 cytosines, 857,000 adenines, and 860,000 thymines. The GC content is 42%, while the AT nucleobase is 57%. So how could we use this in real life? Well, I'll give you two 1 or 2 minute examples here. The first example that we'll use being that we are using the breast cancer data set is going to be an example about disease. So when you go to your physician and they ask you about your parents' background or medical background, they want to know if they've had cancer or if you've had cancer in your family or if you've had diabetes in your family or if you've had Alzheimer's in your family. And the reason they ask these questions is because of the principles of DNA. Whereas if your parents actually had any of these diseases, does not necessarily mean that you will actually get the diseases. However, it does put you at a higher risk given the laws of deoxyribonucleic acid. So that is an example of why your physician asks you about your family background. The second example I'm going to give you about DNA is the GC content versus the AT nucleobase. This is another sequence method where the DNA sequence is actually compared between two people. So it could be like a paternity test or a maternity test. And so this is why it's not good for you to keep it PG, we'll say interact with a significant other's relative, be it a first cousin or better, because not only is it ethically a disaster, it could also be a legal and biological disaster as far as paternity or maternity being established. For example, there's two types of tests. One is an accuracy DNA and the other is a probability DNA. The difference is that a probability DNA test pretty much tests for certain markers that are shared between the two test subjects. So this is to say, well, are they 100% excluded from being related? So you can actually have just 2 or 3 markers in a DNA sequence that match, and that particular test will turn out to be what we call a false positive. And though that is 99%, which is to say, if you are in the room with 100 people, then 1 out of 100 of those people share the same DNA versus 99.999999%, which is to say, if you were to test amongst 1,000,000 different people, only one of those person out of the million would have matching DNA. And that would be the accuracy DNA test. So this is why it's not a good idea to date one's family member, because if it is a probability test, and let's say that a mother dated your father's brother and you did a probability test, and you might show up 3 or 4 markers, and that particular test just might come up 99%, whereas the actual father would show 99.99999%. So that's why it's not a good idea to actually interact with your significant other's relatives outside of the ethics. So that's scientifically speaking. So next we are going to go over Biopython, which is a library that gives us a set of research tools to further do research in DNA. See you there.